Bismillah ar-Rahman ar-Rahim. The title of our practical session is in renal module is ligand assay, and the ligand is due to use a substance that bind either with the antibody or antigen, and this is called the conjugate, and this substance that bind to the antibody either fluorescent material or enzyme or radioactive material. First we have the immunofluorescence test, and it's called so due to use the conjugate, which is the fluorescent material, and it's of two types, direct and indirect method. The direct method, we use the tissue from the patient, like skin biopsy or renal biopsy, that contain the antigen in the skin or in the glomeruli of the kidney, and then we add to it the conjugate, which is the antibody labeled with fluorescent material, and then examined under the fluorescent microscope. And this is used to detect the autoimmune disease in the glomerulonephritis or in the pemphigus vulgaris, in the which is autoimmune disease of the skin, characterized by blisters filled with fluid. While the skin type of the immunofluorescence is indirect type, and we use the biopsy from the animal, which is usually used the rat or mouse, and we took biopsy from the liver, from the stomach, from the esophagus, and then we add to it the patient serum that contain the antibodies, and then we add the conjugate, which is the antibody labeled with the fluorescent material. And this indirect method used to detect the autoimmune disease, like anti-nuclear autoantibodies, anti-smooth muscle autoantibodies. And here the immunofluorescence test, usually it's used the fluorescent microscope, not ordinary microscope, and examine the slide, and then we see the apple green color of the tissue, and this is used to detect the autoimmune disease of the thyroid, and this is the autoantibodies against the thyroglobulin, and here against the thyroid cell. The second type of the ligand assay is ELISA, which is abbreviation of enzyme-linked immunosorbent assay. And here we use the microtiter plate, and this plate will be coated with the antigen, and from the company that synthesized the kit, and then we add the patient serum that contains the antibody, and then we do washing to get rid from the excess antibodies, and then we add the ligand, which is the antibody labeled with the enzyme, and then wash, and then add the chromogen and substrate, and then we get a color which is yellow color, and this is used to detect the antibodies against the HIV, antibodies against the hepatitis B surface antigen. And the principle of the ELISA, the antibody labeled with enzyme, usually use the horseradish peroxidase enzyme or alkaline phosphatase enzyme, and then add the substrate, which is the H2O2, and this leads to liberation of the oxygen, and the oxygen will oxidize the reduced chromogen, and then we end with oxidized chromogen and water. And this is H2O, not H2O2, water, H2O. And here the plate of the microtiter plate of the ELISA, and each well, which is for one patient, and read the color by spectrophotometer or ELISA reader, and we get the concentration of the antibody, and it is the quantitative method give the concentration of the antibodies in the serum. The another test, which is a RIA, which is abbreviation of the radioimmunoassay, and here we use radioactive material instead of enzyme. It's the same principle as ELISA and previously used for the hormonal assay. Nowadays no longer used because of its difficulty to get rid from the radioactive materials that used in the test. And the another new test, which is immunochromatography, which is a qualitative and rapid test using a dipstick membrane coated with monoclonal antibodies against the antigens. And here the conjugate, which is a protein A colloidal gold conjugate, and using in many tests like detection of malarial antigen and the pregnancy test, and nowadays used to detection COVID-19 virus by detection the antibodies against this COVID-19 antigens. And it is a rapid test within few minutes, and it's used to early detection of the infection. And it's used either to detect the IgG, which is for the secondary immune response, or IgM, which is for the primary immune response that indicate recent infection, or the detection the IgG that indicate past infection. And here the immunochromatography, which is used for the pregnancy and nowadays for the COVID-19, and used for the malaria, used for the process-specific antigen, used for the viral infection. And this is very easy and done, and we got our results within minutes. The another test, which is old test, and it's used or also for detection antigen or antibody, and it's called complement fixation test. And here we have two tubes. Here we add the antigen, and here we have the antigen, and then add the patient serum to each tube. If the patient is infected, so will develop antibodies against these antigen. If it's not infected, so there is no antibody against these antigens. So this antigen antibody immune complex, you cannot see this reaction. So add to it the complement, and this leads to antigen antibody complement fixation, while here the complement will remain free. And then we add the indicator cell, which is called receptor cell, and to see the reaction between the antigen antibody and the complement. And here, when the complement consumed by this antigen antibody immune complex, so the receptor or indicator cells or sensitized RBC will remain free, while here, when there is no antibodies, the antigen and antibody will remain, the antigen and the complement remain free, and the complement will bind to sensitized cell, lead to destruction of the RBC and release the hemoglobin. And the indicator cell or receptor, which is consist from the RBC coated with the antibodies. So when there is rupture of the